Hi, my name is Kai. I'm from M3TRIQ. Our idea is called AMPSA, and we are joining the track of agentic automations. AMPSA stands for Agentic, Multi-Objective, Paratope-preserving, Species Adapter. Essentially, is a universal antibody adapter from one species to another one. So antibody adaptations is very well practiced in modern medicines, because often we develop treatment in mice, antibody treatments. We have to humanize it in order to run it in human clinical trials. Otherwise, humans or the target species will be allergic to the hostile different host antibody frameworks. This process takes months and costs millions of dollars, and is usually have a high failure rate. And we have been practicing it manually so far. So I think it's about time we can use it, use AI to package the workflow. The applications of AMPSA is immense. First of all, it greatly accelerates preclinical trials. Also, second, we can quickly adapt the antibody solution from one species to another one. For example, lucivetimab. Lucivetimab is to treat itchiness in human originally. Later is used to adapt it to dogs. And now with AMPSA, we can immediately adapt this antibody treatment to all the species. Why not treat cats with lucivetimab, and also treat cattle, fish, whatever livestock, or any other species that, for example, endangered species. Because endangered species, they don't have huge numbers, and usually there is less economic purpose to develop antibodies for them. So that can benefit tremendously from our existing library of antibody treatments. Well, of course, the R&Ds will greatly benefit from cross-species antibodies as well in staining and diagnostics. Underneath AMPSA, what is we have 4 agents. So the user input the heavy chain and light chain sequence, and then tell the source and the target species. So, and then the agent will start working. So analyzer agent will first understand the structures, use ANARCI to identify the CDRs. And then next, the designer agent have parallel LLM calls using DeepSeek. It will approach different strategies of adapting the sequence to the target species. And then it use a ESM2 to embed the adapted sequence, to check whether it's how far it is from the centroid. So it's like how far it is from the typical target species antibody sequence. The further it is, the less likely is adapted to that species. And then we filter off the candidates from there. The number 3 is we use a validator agents. Validator agents consist of 5 different scores, where we score for their stability, their likeliness of being expressed, and then how similar are they in terms of 3D structures of the bindings compared to the original ones. And also the last one, we also score for the LLM reasoning quality, because the better strategy will usually generate a better, more high affinity and good antibodies sequence in the end. Last but not least, we use optimizer agents to optimize the sequence and to score, to check whether we have to iterate the entire process. The more we iterate, the better it is to adapt to the target species. And if 1 or 2 rounds, given 1 or 2 rounds, it will be adapted to the species in certain confidence, and it will list out the modification required and also the quality of the adaptation. This is like in one flow, we can adapt a species antibodies. Now we're moving to the front end, where the user will be interacting with the agentic workflow. This is the input fields. For demo purpose, I will show you CompPath, where we humanize a rat sequence CompPath to human. It has been already done, so this is what we try to verify whether our system aligns with what is happening out there. And here is a modified sequence, and this is a 3D structure of it. And here is a list of rationales how the AI approach to adapt the human sequence. And then in the end, there are scores, and on the side there are methodology of how the flow works.